In the previous class, we were talking about what causes gene differential expression, and we told you some examples, some molecular examples. Now we're going to focus on more cellular examples. So here you can see in developmental biology, we're trying to see um, different perspectives. So from the organism perspective, at the cellular perspective, other cell perspective, and from the environment. So in this section, we're going to play with all these factors, how they interact. So for example, how other cells, the signals from other cells will impact the cell, how lone signals will impact, and how external environment. And we will show you that in addition to the genetic components, we have all other aspects that are influencing gene expression. So let's start with this. Here you can find some literature from your textbook and from recent articles in case that you're interested in some of these topics. And from a developmental biology perspective, the history of the interactions that cells have, so previous interaction, the current interaction, will determine the gene activity. Okay, so we are going to talk about juxtacrine interactions, paracrine interaction, the interaction with the environment, and how they create different gene expression. And maybe in cell biology or in anatomy and physiology, you have here different type of signals. So here that are signal between cells, that there are signals that between cells that are very close, and also signals of cells that are very far and they are traveling through the bloodstream. So we're going to cover each one and give you a little bit more of an example of some of those ones. So we're going to start with cells that are close to each other. So in addition to the external environment, cells have an internal environment that it could be the cellular environment. And this can affect, for example, the gene regulation. Yeah, and for example, here you have two cells, cell number one and cell number two. And here you have a receptor, and this one will have a ligand. Well, generally you have ligands that are traveling. In this case, the ligand is not free. Instead, is bound to the membrane. Yeah, so here this type of is called juxtacrine signaling and occur between neighboring cells, and there is a physical contact. And remember, the signaling molecule is not free. Is not free. It is bound to the membrane of the other signal. And one example that I want that you remember is delta and notch signaling. Yeah, so remember it's a short range signal, and delta and notch is found in so many functions, and I'm going to cover a few of them, but there are many. Okay, so here you see a picture where it's a little bit more details what is the delta and notch. That is much more complicated that I showed you in the previous slide. And rather than I'm not going to ask you how many domains of protein they have. I'm not going to ask you the details of the delta and notch, but I want that you remember that this is a highly conserved signaling system, okay, present in most animals. So for example, in fruit flies, the production of bristles require delta and notch. Very important. It is important also for cell communication and involves gene regulation mechanisms that control cell differentiation. So I told you that it was important for fruit flies, but it could be also important for tumorigenesis and for so many things that, so we use fruit flies as an example, as a system to study cancer because the same delta notch is expressed in both. Now I'm going to give you some examples of the pleiotropic effect of notch signaling. So here I took this from this paper, and here they summarize for examples. So for example, in the self-renew one, so you have stem cells here, and notch signal is in charge of regulating the self-renewal of tissues. That is very ironic because it's also when it's unregulated, it could be also in charge of tumorigenesis, the production of tumors. So Notch maintains the stems and or TA cell. These are here. You see the TA cells in an undifferentiated state. Another function that is common in between Notch signal is that is able to define the binary cell fate decision. So between, for example, an epithelial cell and a basal cells in fruit flies, and the same thing in other type of tissues. So NOx is going to inhibit one, yeah, and promote the development in the other one that is not expressed. And this is how you can have this potent cell fate. Okay, so Notch is very good at doing these kind of things. Finally, another function of Notch pathway is the induction of differentiation. So, for example, in the skin, Notch induces the terminal differentiation. So you have these again TI cells that are cells that are going to differentiate very rapidly, and Notch is able to speed up this or proceed to the differentiation. Yeah, so for example, Notch 1 promotes the differentiation from proto T cells into pre-T cells. And as I said before, Notch can be considered an oncogene and is involved in the process of tumorigenesis. It has been shown that the overexpression of Notch can be involved in the development of some leukemias. In the skin, Notch can have the opposite function. In this case, it will be a tumor suppressor. So the loss of function result in carcinoma-like tumor suppressor. And this is very common in Notch. You can have one function in one tissue and another function in another tissue. And these are just few examples to show you that Notch can be involved in a lot of things. Okay, so now let's move into the second example, that is the paracrine cells. Here the cells are close, but not as close as before. So do you remember this diagram that I told you that the cells have? We generally think about the organisms that the environment is outside, but there is another environment that is the internal environment, and even this inside of the cell that is also a cell environment. Now we're going to talk about the paracrine signals, that in this case you have diffusible factors that they are not as close as before to the cell. They are not linked together. There is not a physical connection, but the factors are relatively close, and they can be diffused to the other cell to have a difference in gene expression. Okay, in this case, the Parkin functions, they have three main characteristics that I'm going to tell you in this slide. Yeah, here 1, 2, and 3. The first one is that the signals act locally between the cells, and they are close together, as I said before. The second thing that I told you is that they move through diffusion in the extracellular matrix. And finally, this type of signal usually produce quick response that is usually happen for a short amount of time. So it's opposite to what happened with the hormones that are far and they need to travel and they can produce an effect that lasts for a long time. These paracarrier signals have the opposite function, but in both cases they can have changes in the gene regulation. So we already talked about the juxtacrine, and here is delta notch. Uh, and I just told you about the hormones and the endocrine signal very quick. There we will give you an example about that. There is a long range signal, and now we're in the paracrine signal. And look, remember Wnt pathway and the BMP. So here, these ones are local signals, are paracrine signals. Okay, so one example of a paracrine signal is, you can see it here. Here is the line again, that is BMP, or bone morphogenetic proteins. And they are common growth factors. They have also other functions like pinocytokines and metabolic genes. I'm not going to ask you about those things. What I want that you know is that there is another pathway involved into that. And here are some of the components. There is usually a receptor. And here you will hear in other cases all of these different components like SMAD, that they will end up going to the after triggering a cascade of events, they will do a change in the gene expression. Okay, so again, these diffusible factors will end up changing the gene regulation, and this is why it's very important to study them because this is another mechanism of changing the gene regulation during development. In fruit flies, we don't have BMP. We change the name. We give them the name DPP. That is decapentaplegic. Very easy to remember. I'm not going to ask you to remember that name for the test. What I want that you know is that this is the fly homologue of BMP. So sometimes you can find one molecule in one organism and another molecule in an organism until you finish all the pathway. You recognize that they were highly conserved and they were relatively similar. So this is why sometimes we change the names, but those molecules are homologues. Okay, so we just finished talking about physical context. That it was the one you have juxtacrine functions. Then we talk about paracrine function, that is diffusion. And now we're going to talk about the external environment, how we can change the gene regulation. Okay, so let's start doing that. To finish this section, I'm going to talk about the external factors, in this case the diet and the sunlight, and use the production of the vitamin D as an example of how the gene regulation require of external factors, and changes in the external environment can have changes in the gene regulation. Okay, so to do that, this is the big picture and this is the most important part. Yeah, and we are going to go into a little bit of the molecular aspects, but the key idea is here that the sunlight, your diet, and even supplements are important to produce vitamin D3, that later it will be processed in your liver by a series of pathways that we will talk, and the kidney, and it will go to the bloodstream and produce a lot of changes, and as a result, you will have healthy muscles and healthy bones. Okay, so let's start with that. So let's go into more detail. Let's zoom in, and the first step, as I told you, was the sunlight exposure that go into your skin to produce vitamin D3. Then the diet also is another source of vitamin D3 and D2 that both end up going into the blood vessel and to the liver. Here we're going to, you don't have to memorize all the chemical structures or all these names that are here, but we're going to talk a little bit more in the following slides. Then the four is that these compounds go to the liver and then to the kidney and then uh, go inside of the cell. This molecule called calcitriol, that is the one calcitriol that is going to be the one that change the gene expression. So, and everything starts here from the sun or the diet. So the external environment play a key role in the regulation of gene expression, and is one of the reasons why we have the same genes but different phenotypes. So what happened here in the beginning is that for the synthesis of vitamin D3 occur in the beginning in the skin, where here you have, as you can see here, pro vitamin D3 that is converted in pre-vitamin D3. Yeah, in response of the sunlight exposure. And this is the formula. I don't want that you remember, there is no need that you remember this, but there is a process of photolysis, something similar to what happened during photosynthesis. And here there is end up with the production of the vitamin D3. Course, the process is more complex, and for example, in the epidermal basal layer, there is the isomerization of the vitamin D3, while in the other part of the pathway, in the diet, you can have intestinal absorption of the supplements D2 and D3 that are going to end up binding to the binding protein, or DBP, that will be located in the bloodstream. So two different sections, two different ways to get vitamin D2 and D3, sunlight and diet. And again, here are the different names of the molecules. If you want to know them, I'm not going to ask them in the test. What I want that you see is the following, where you see that it goes from the liver to the kidneys and it goes to the hormones. If you remember, this is another way to produce a change in gene expression, that the hormones are have different from the paracrine signal. This is a long distance and more prolonged changes in the gene expression. And so they will go in the bloodstream and they produce this calcitriol that enters to the cell that it will change the gene activity. Okay, so far, this is the key information that I want to show you now. As usually, I am going to show you a little bit more of the molecular basis of what is happening there. You don't need to memorize all the steps. I just want to show you that, again, the same things are happening. Well, you have a receptor, then there is a process of transduction, and later on in the cell, there is a change in the gene expression or the gene activity. So here you have the calcitriol that is going inside of the cell, that is going to, as it goes inside, in this case, it is going to go and attach to this specific gene and produce changes in gene expression. Again, you need to remember the cofactors, the coactivators. I just want that you remember that that the external factors are the ones who lead this process. So one thing that maybe you remember, here the receptor is not in the membrane, here the receptor is in the cytoplasm. This happens when with hormones or with molecules that have a lipid origin or that can cross the membrane, so they don't need to have a receptor. So here you have the vitamin D receptor in the cytoplasm that is going to attach to this REXR, retinoic acid receptor, that is going to be at the DNA and is going to uh, produce a change in the gene activity. Okay, so again, this part could be more complicated as I showed in the previous slide. I just want that you remember uh, uh, just the basis of this, and I just wanted to show you that not all the receptors are going to be in the membrane. Some of them are going to be in the cytoplasm, but it's the same idea. They attach to the receptor, and later there will be a transduction of the signal, and later on there will be a change in the transcription or changes in the gene activity. Okay, so with this we finish this section, and we talk about how the external environment, diet, and sunlight can affect the gene activity, and in this case, it can make your bones and muscle healthier by the production of vitamin D3. So we came back again to this slide when we compare local signals with long-range signals, to physical contact, to diffusion factors, to external factors, and all of them have an effect on the gene regulation. And with this, we also finish the different formats of signals that we discuss, from the ones that are very close to the ones that are very far. Okay, the only aspect that we miss from this class is the cytoplasmatic factors. Yeah, so we have talked about other environment except for the internal cellular environment. And a beautiful example that shows that the cellular environment is important is the animal cloning. Here we have Dolly again, and you can see here that they took the cell from a mammary gland, and you see the nucleus, yeah, in red, yeah, but also you see the cytoplasm in yellow. So if you see from this nucleus is competent, yeah, is able to produce a mammary gland cell from a specific genes that are located there. So so at this point, this is following the postulate of the genetic equivalence. But now let's see what happened later. What we did later is we put the same nucleus here, and we remove the nucleus of the oocytes. And now what is happening? Now the nucleus is being competent, yeah, to activate early developmental genes. Yeah, so the cytoplasmatic factors, the internal environment, is also very important. Yeah, it dictates to the nucleus what it's supposed to do. That in this case is to produce a new organism, not a mammary gland like in the beginning. Why? Because the internal environment of the cell is also very important uh, for the gene regulation. Okay, so with this, we're done. We know that although we have same genes in developmental biology, we can have different environments, and I show you multiple cases of environments from very small ones like the internal environment of the cell to large environments like the sunlight that we receive. And I show you this can lead to differential gene expression and different cell fates. If you have any question, just let me know and have a good day and bye.